Am I audible?
Yes,
Hello?
ma'am, you are. Yes, ma'am.
Okay. Yeah. According to Food and Agriculture Organization, by 2050, world's population will be 10 billion. And to feed this huge population, we need to produce our food, need to increase in the production of our food by 100%, which is a huge, huge percentage. So the world's food production and distribution are facing great stress. I can show you the data. Today, agricultural sector is using staggering amount of resources, just such as 187 million tons of fertilizers, 4 million tons of pesticides, 2.7 trillion cubic meters of water, which is about 70% of our all fresh water, and huge amount of thermal energy, okay, for global annual food production. But still there are approximately 850 million people are currently undernourished, and it is expected that by 2050 there will be 2 billion people in this category. Okay. Therefore, the situation calls profound changes in the global food production system, and therefore sustainable agriculture is very crucial to achieve zero hunger in the country. Therefore, my whole presentation is divided into two parts. Part 1 contains nanotechnology. Here we'll see what are nanoparticles, then how nanoparticle impacted agriculture and human health, then strategies to improve agricultural production. Part 2 contains zinc sulphide nanoparticle. Here we'll see why we have chosen zinc sulphide nanoparticle, then its synthesis and characterization, and then its application. Now, let me tell you a short story about nanotechnology. The term nanoscience and nanotechnology started with a talk entitled "There's Plenty of Rules at the Bottom" by a physicist, Richard Feynman, in 1959 at annual American Physical Meeting. In his talk, Feynman predicted that there will be a time when we will be able to make things at atomic level. Feynman considered the possibilities of direct manipulation of individual atoms as a powerful form of synthetic chemistry than those used at that time. The version of his talk was reprinted in a few famous journals, but largely it was unnoticed. Over a decade later, Professor Norio Taniguchi coined the term nanotechnology in 1974, but it was not yet. After development of scanning tunneling microscope, which could see individual atom, modern nanotechnology began in 1981. So what is nanoscience? Nanoscience is a science, engineering and technology conducted at nanoscale level, which is around 1 to 100 nanometer in size. A nanoparticle is so tiny, so small, it is hard to imagine. Okay, 1 nano means 1 billion. And 1 nanometer means 1 billion part of a meter. If we assume a brick or a marble as a nanometer, then 1 meter will be the size of our Earth. It is so tiny. Now, this is a scale bar which indicates the sizes of different objects. At first, we have 1 millimeter to 1 micrometer scale bar where we have hair cells, red blood cells, bacteria, chromosomes. And in nanometer range, we have genes, viruses, ribosomes, antibodies, molecule, atom, DNAs. So we can get an idea that how small, how tiny nanoparticle is. A nanoparticle has different important properties such as size, shape, surface functional properties, material compositions. Okay. There are different shapes for nanoparticle. They can be sphere, cube, rod, star-shaped plate, but most of the time sphere nanoparticle are used. At nanometer level, we can manipulate these properties and we can train them, we can make them behave as we want it to do. Modern nanotechnology is a very new field, but different size nanoparticles was used for centuries. For example, 100 years ago, nanosilver and nanogold particles were used in stained glasses in the medieval churches. But back then, artists didn't know the process. But now, as we know how to synthesize nanoparticles, we can make them according to our need, according to our target agent. Okay. Therefore, scientists are finding different ways to synthesize nanoparticles to achieve the enhanced properties of nanoparticles, such as great stability, strength, great chemical reactions, great control in light spectrum in comparison with their large form material. Now, does this nanotechnology has place in sustainable agriculture? The question is yes, it has. Sustainable agriculture can be explained as integrated system of plant and animal production practices having a site-specific application such as land farming, and the long way uh, and its main target is to improve human food and fiber need, essentially improve the life of human overall for farmers and rest of the society as a whole. Nanoparticles and nanopesticides can improve crop productivity. A nanoparticle can provide missing nutrient to the plant. For example, zinc deficiency has been long noted as most limiting factor for the crops, but this problem can be solved by the application of zinc nanoparticle. Nanoparticle can also improve the quality of soil. For example, nanoclay can improve the water binding capacity of the soil, okay, which can reduce hydric shortage during the crop season. It can be act as nanosensors to monitor health of soil as well as plant and ultimately stimulate plant growth. We have different strategies to improve crop productions, like we can transfer genetic material through nanoparticle and form transgenic plant. By nanopesticides, we can protect crop. By nanosensors, we can monitor its plant and soil health, and we can make plant stress tolerance too by the application of nanoparticle. As I have told in my previous slide, that nanoparticle can enhance soil fertility. It can enhance crop growth also. Mm, As nanoparticles are used in agriculture, it can be toxic to non-targeted organisms also. So before doing any agricultural or field-related experiments, one should keep in mind the toxicity part also. Now I'm summarizing the points. Today, nanotechnology is an emerging field of science with far-ranging applications such as cancer therapy, targeted drug delivery in biomedicines, wastewater treatment, cosmetic industries, electronics and biosensors, etc. Global investment in the field of nanotechnology has been increasing worldwide. There are thousands of commercial products are using nanotechnology or nanoparticle to enhance product quality and their stability. But this huge application of nanoparticle could lead to their accumulation in ecosystem and entrance into the food chain. Okay. Therefore, as a novel and emerging class of products, the ecological risk of nanoparticle is an important topic. Now, plants play a crucial role in transportation of nanoparticle. A number of studies have shown the phytotoxic effect of nanoparticle at certain concentration by producing huge amount of reactive oxygen species. After accumulation inside the plant, they can start degrading the crop quality by decreasing seed germination, photosynthesis, overall biomass, etc. A nanoparticle can interact with soil organisms. Okay, Here you can see nanoparticle can interact with soil organisms which can facilitate its entry inside the plant vascular system through symplastic or apoplastic pathways, and once it is inside the vascular system, it can go inside the plant cells through different mechanisms like endocytosis, pore formation, through carrier protein, plasmodesmata, etc. Now, as plants are primary uh, producers, so it can serve as potential pathways for the transportation and accumulation of nanoparticle into food chain. Nanoparticle move via pore, but the movement of nanoparticle via pore is size specific. Plant translocate from root to shoot and accumulate in the different parts of the plant. Okay, now plant can also activate various enzymatic and non-enzymatic defense system against the nanoparticle induced stress. But extremely high concentration of nanoparticles were used in most of the toxicological studies, which might inhibit the potential beneficial role of nanoparticle. For example, there are many studies which show the beneficial properties of nanoparticle as well. For example, in 2013, Pradhan et al. Reported that metallic manganese nanoparticles were a better micronutrient source of manganese than the commercially available manganese salt. Mm, So, part 2, zinc sulphide nanoparticle. It is a main source of zinc in nature. It is widely used semiconductor. It has wider band gap, easy to synthesis, good biocompatibility is there. And it is extensively used in rubber and paint industries, which aggravates its chances of leakage into the environment. It is generally non-toxic in nature, and there are few studies too which shown the beneficial role of nanoparticle in the plant system. For example, zinc sulfide nanoparticle at 15 ppm concentration increase the growth of brassica plant. Therefore, the objectives of my studies are synthesis and characterization of zinc sulfide nanoparticle, studying the effect of zinc sulfide nanoparticle on plant growth and antioxidant defense system, and finally, understanding the mechanism of uptake and translocation of zinc sulfide nanoparticle by plants. We have synthesized zinc sulfide nanoparticle by chemical precipitation method, and this is the formula for synthesis of zinc sulfide nanoparticle. And after synthesizing zinc sulfide nanoparticle, we have characterized the particle with UV spectroscopy, XRD, FTIR spectroscopy, PL, scanning electron microscope, EDX, and transmission electron microscope. This is an image of UV spectroscopy. This is showing the characteristics hump-shaped curve for synthesized zinc sulfide nanoparticle, and confirmed that the synthesis of zinc sulfide nanoparticle was successful. This is a XRD image of synthesized nanoparticle, and these are the characteristics of synthesized zinc sulfide nanoparticle. Now this is a FTIR image which shown the surface functional group of synthesized zinc sulfide nanoparticle. And finally, this is PL spectroscopy image of synthesized nanoparticle, and confirmed that the synthesized nanoparticle was zinc sulfide nanoparticle. Because of these are the characteristic for zinc sulfide nanoparticle. These are same scanning electron microscope and transmission electron microscope image of synthesized zinc sulfide nanoparticle, which confirmed that the synthesized nanoparticle were nearly spherical in shape and below 20 nanometer in size. Okay. Now, objective 2, effect of zinc sulfide nanoparticle on plant growth and antioxidant defense system. To understand the effect of zinc sulfide on plant growth, we have chosen moonbeam plant as a model system and treated moonbeam plant with a different concentration of zinc sulfide nanoparticle. Those are 0, 0.1 mg per liter, 0.5 mg per liter, and 1 mg per liter concentration, and found that the treated seeds have significant increase in germination percentage than the control. Now, our entry of ions inside the seed is a tough task because of hard coat of the seed. But in case of nanoparticle, due to its tiny size, it can easily enter into the seed through water transport movement and enhance the seed germination. Again, nanoparticle can form pores which can facilitate the nutrient uptake inside the plant. So, in our study, zinc sulfide nanoparticle treatment significantly increased seed germination in treated seeds. This is an image of 10 days old moonbeam plant treated with different concentration of zinc sulfide nanoparticle. These are hydroponically grown moonbeam plant and showed that zinc sulfide treated nanoparticle were healthy and significant increase was found in root and shoot of treated plant in comparison with the control. This result confirmed that the zinc sulfide nanoparticle has positive impact on root shoot length of moonbeam plant. Now, to study the uptake of nanoparticle by plant, we have measured zinc content inside the plant tissue and found that significant increase in zinc content found in root and shoot of treated plant in comparison with the control, which again confirm the uptake of nanoparticle by the plants. Now, photosynthesis is a very sensitive process and its efficiency decreases under stress. To investigate the effect of zinc sulfide nanoparticle on photosynthesis, we measured pigment content and found that treatment of zinc sulfide nanoparticle significantly increased pigments in moving plant. Okay. In plant, chlorophyll A and chlorophyll B are the most important pigment responsible for photosynthesis. Chlorophyll A is directly associated with the photochemical reactions, while chlorophyll B indirectly helps in photosynthesis by transferring energy to chlorophyll A. Again, chlorophyll A is directly associated with the carbohydrate production, which is responsible for better health in plants. Therefore, increasing chlorophyll A and chlorophyll B pigment indicated the positive role of zinc sulfide nanoparticle on plant photosynthesis. Okay. Carotenoid pigments are the light harvesting pigment of plant and it absorbs light and transfer energy to chlorophyll A. It protects chlorophyll from oxidative stress. So, increase of carotenoid pigment by the treatment of zinc sulfide nanoparticle also confirm the positive role of zinc sulfide nanoparticle in augmenting plant photosynthesis growth and protecting role of zinc sulfide nanoparticle in electron transport chain. Okay. Now, hydrogen peroxide plays an important role in plant stress mechanism. In optimum concentration, hydrogen peroxide act as signaling molecule involved in triggering and signaling cellular growth. But at high concentration, hydrogen peroxide induces oxidative stress and programs cell death in plant. In our study, you can see there was no significant alteration found in any treatment. Okay. And the peroxide content in both leaf and root were in optimum range, which indicated that due to zinc sulfide treatment, peroxide content remains in optimum range and it acts as signaling molecule for the plant growth. This could be a reason for enhancement of growth in moving plant. Now, an interesting result was found with liquid peroxidation. We found a significant decrease in lipid peroxidation in the treated plant. Lipid peroxidation is an indicator of membrane integrity and damage. So decreasing lipid peroxidation by zinc sulfide nanoparticle confirmed that it acts as protective uh, for membrane integrity. Now, proline is also an important marker for plant stress management. Um, we can see there is no significant alteration found in leaf proline content, whereas significant decrease found in root proline content, which again indicated the positive role of zinc sulfide nanoparticle. Now, electrolyte leakage. We have found significant increase in root electrolyte leakage. We can say that as roots were in direct contact with the nanoparticle, there may be a little increase in electrolyte leakage happen, but I'll explain it in my later slides. Now, role of zinc sulfide in antioxidant defense system. Um, In our study, we did not find any change in phenol content, whereas there were significant increases found in flavonoids content, total antioxidant activity content, DPPH activity content. The increase was in dose-dependent manner, which indicated the antioxidant attribute of zinc sulfide nanoparticle. Now, the role of zinc sulfide nanoparticle on ROS generation. Nanoparticle can produce huge amount of ROS and generate oxidative stress inside the plant. But in our cases, none of the treatment produce any kind of oxidative stress. This is superoxidase dismutase enzyme. This is a powerful enzyme of plant which catalyzes oxygen radicals to hydrogen peroxide. And we have found that at 0.1 mg per liter concentration of zinc sulfide nanoparticle, the superoxide dismutase activity decreased. Okay, this decrease indicated the antioxidant attribute of zinc sulfide nanoparticle, and with the catalyst activity, we have found there was significant increase in root catalyst activity. This result was correspond with the electrolyte leakage, as there were also electrolyte leakage increase in roots as well as catalyst activity increase in roots. These results confirmed that the organs which comes in contact with nanoparticle directly can enhance catalyst activity. Okay. But the range of increase of catalyst activity and electrolyte leakage were in optimum range, and this range is not enough to produce any kind of oxidative stress inside the plant. Now, we did not find any significant alteration or difference in the peroxidase, and ascorbate activity, but we find significant increase in leaf glutathione reductase activity. Previously, it was reported that plants with decreased glutathione reductase activity are more prone to stress. They are smaller in size. So here in our study, glutathione reductase activity increased in the treated plant, which signified that zinc sulfide nanoparticle can increase glutathione reductase activity and also responsible for increased growth in plant shoots. Now we'll understand the mechanistic approach of uptake and translocation of zinc sulfide nanoparticle by plant. This is a scanning electron microscopy image of control and treated plant root and leaf. This image is showing that the control root and treated root are similar, There are no significant alteration found in the control and treated root, um, as well in leaf. There are no significant changes found in the cellular structure of the treated plants in comparison with the root, which again confirmed that zinc sulphide nanoparticle did not enhance any stress inside the plant. Now, this is the EDEX analysis of control and treated plant. This is the EDEX report, which is slightly blurred right now, not showing clearly. But this is the EDEX analysis of treated and controlled plant, which confirm the uptake of zinc by the plant. Now, this is the 10 images of leaf which confirm the uptake and translocation of zinc sulfide nanoparticle. These are the control leaf section. As we can see, there are no particle present in the vacuoles of the plant leaves, whereas in the treated plant there are these nanoparticles are present in the vacuoles, which confirm the uptake and translocation of synthesizing sulfide nanoparticle from root to leaf. This is the root section of control and treated plant. Again, confirm the translocation of zinc sulfide nanoparticle from root to leaf. Now, uh, nanoparticles are not always toxic. Dose of nanoparticle, plant species, etc. play vital role in the toxicity. In our study, zinc sulfide nanoparticle treatment promoted root shoot length and produced higher photosynthetic pigment in treated plants. None of the treatment trigger any kind of oxidative stress but improve antioxidant system of the treated plants. The electron microscope studies confirmed uptake and translocation of zinc sulfide nanoparticle in treated plant and also showed that it did not cause any damage to the cellular microstructure. Uh, there was no nanospecific toxic effect on plant growth found, rather displayed some positive changes, and this finding needs to be considered in environmental risk assessment and management strategies. But further studies will be necessary before using zinc sulfide in the field. Future perspective. Micronutrient malnutrition has affected lives of billions, with about 5 billion suffering from iron and 2.7 billion suffering from zinc deficiency all over the world. A striking figure of 27% of total population in India is affected by zinc deficiency related to disorders such as poor immune system, diarrhea, poor physical and mental health. Therefore, zinc deficiency claims about 4.4% of the total child death in the world. Therefore, even a small increase in the nutritive value of crops can be highly significant for human health and nutrition. For this, biofortification has emerged as one of the possible solutions to alleviate malnutrition, and hence zinc sulfide nanoparticle can be used as zinc source in agriculture, but more systematic research will be required in future to develop fortified foods. I'd like to thank my supervisors, Dr. Prashant Kumar Bishash, Associate Professor in Food Technology, Biochemical Engineering Department, Jadavpur University, and Dr. Abhishek Mukherjee, Associate Professor, Agricultural and Ecological Research Unit, Indian Statistical Institute, for their invaluable advices and continuous support and encouragement throughout my research work. And I sincerely thank Dr. Chandan Kumar Ghosh, Assistant Professor in School of Nanotechnology in Jadavpur University, for his stimulating suggestions and encouragement that helped me to coordinate my project, especially in writing part. I thank all the faculties of Food Technology and Biochemical Engineering Department in Jadavpur and Agricultural and Ecological Research Unit in Indian Statistical Institute for their valuable suggestions in every instances. Now, this is my pleasure moment to thank all my colleagues, lab mates, and friends for their continuous help and support. I would like to thank all the technical staff of Jabalpur and ISI for their support and help in experiments. Okay. It is my great privilege to thank all those who have directly or indirectly helped me throughout the experiments. I specially thank ISI TAC project for the financial support during my research, which made smooth research. And last but not the least, I would like to thank all the members, all the team members of Biological Club of IISER Thiruvananthapuram for giving me this opportunity to present my research in this wonderful platform. This is my lab group. This is my ISI lab group. Okay, and this is my research paper. If you have any further questions, if you want to know some more details, you can go through this paper. These are the references. And finally, thank you all for your attention and patience. Thank you.
Thank you very much, ma'am, for a highly intriguing talk. Now, I welcome everyone to ask your questions. Please use raise hand feature to do the same. You can also type out your questions in the chat box. So there is a question in the chat box. You had mentioned earlier about using nanoparticles for pest control. Is there any way that this could affect humans as well?
You had mentioned earlier about using nanoparticle for pest control. Okay. Is there any way that could affect human as well? Uh, the question is little bit confusing. Who's asking this? Vidya Rashmi. Are you asking in a toxicity aspect or what? Actually, अच्छा, toxicity aspect, yeah. Uh, that pesticides is itself a chemical, right? Which is the overuse of pesticides can affect non-targeted organisms from animals to humans because ultimately it comes into the human food chain. So it is very important to reduce the use of chemicals, and by using nanoparticle we can reduce the effective concentration. Like I show uh, as I showed you that huge amount of chemicals right now using in agricultural sector. So by using nanoparticle, we can reduce the effective dose. So we can and also we can do targeted delivery with the nanoparticles, so we can reduce the effects to non-targeted organisms by the use of nanoparticles. Okay. Hmm. Who's asking now? Dev Mallo. Acha, Dev Mallo. Dev Mallo. He is from my laboratory in Jadavpur University. He is asking. It was really a okay. Okay. No, he's not asking. He's just appreciating. Okay. Thank you, ma'am. Okay. Welcome, Vidya Rashmi. Okay. Anybody else?
There is one more question in the chat box.
Shreya, yeah, Shreya. Apart from zinc sulphide, are there any other compounds of zinc that could be used as fertilizers? Why zinc sulphide in particular? Okay, this is a very good question. Okay, this is a very good question, Shreya. Right now, agricultural sector is using two kind of zinc source. Those are zinc sulfate and zinc chloride to some extent. And in case of nanoparticle, agriculture is using zinc oxide nanoparticle. Okay, but there are very limited reports with zinc sulphide nanoparticle. Very less report are there for zinc sulphide nanoparticles. So it is important to see that zinc sulphide nanoparticle can be a source of zinc can be also a source of zinc. That is why we have chosen zinc sulfide nanoparticle. Like zinc oxide nanoparticle, zinc sulfide also used drastically in industries, but zinc sulfide nanoparticle is less studied. So we have chosen zinc sulfide nanoparticle. If there is any good positive role for zinc sulfide nanoparticle, we wanted to test that. For that, we have chosen zinc sulfide nanoparticle. Okay, Dave Mando again asking that have you done any plant tissue toxicity study to understand that whether zinc sulfide is really less toxic or it is really is it already a known fact? Plant tissue toxicity. Plant tissue toxicity means, Dave Mando, you are asking about tissue culture, plant tissue culture. Yeah. Okay, I did not do any plant tissue culture toxicity study, but there are many papers are there which have done with plant tissue culture and nanoparticle toxicity study. You can go through those papers. And in case of zinc sulfide nanoparticle, I mainly focused on its physiological impact as well as toxicity in plant through plant biochemical assays, and I focus mainly on transport mechanism, uptake, and overall plant growth. Okay, so I didn't get any toxic effects on overall growth on non-green plant by using zinc sulfide nanoparticle. Okay, anybody else? Welcome, Devmando, welcome, Shreya.
So, if there are no more questions, I would like to conclude by thanking Ms. Mala Thapa and everyone present here for making this talk a great success. I would like to thank
It's
our
my
sponsors
pleasure.
once again. I would like to thank our sponsors once again. We are also very grateful for the continued guidance and support of the School of Biology and our faculty advisor, Dr. Sadananda Singh, who made this symposium possible. Looking forward to seeing you all tomorrow for another set of amazing talks by Professor Veena Vaswani and Mr. Gregory Yudan at 4:30 and 6:30 p.m. respectively. Thank you.